everyone, welcome to another video of the Science Club. In this video, let us discuss about the bacterial growth curve. In laboratory conditions, bacteria are usually maintained on a plate known as the Petri dish or the Petri plate. With the help of a sterile loop, bacteria can be inoculated in a liquid culture medium. Bacteria, when inoculated in a liquid media, begin to multiply and increase in number. Since bacteria are unicellular organisms, they tend to reproduce asexually by the means of a process known as binary fission. Bacterial growth is the increase in the number of bacterial cells rather than the increase in their cell size. The growth of these bacterial cells takes place in an exponential manner, that is, one cell divides into 2 cells divide into 4 divide into 8 convert into 16 gets converted into 32, and so on. This process is known as the binary fission. Bacteria require certain conditions for growth, and these conditions are not the same for all bacteria. Factors such as oxygen, pH, temperature, and light influence microbial growth. So, what exactly is a bacterial growth curve? In nature, bacteria do not experience perfect environmental conditions for growth. Therefore, species that populate a habitat change over time. In a laboratory, however, optimal conditions can be met by growing bacteria in a closed culture environment. When grown in a liquid culture, a predictable pattern of growth in a bacterial population occurs, and this pattern can be graphically represented as the number of living cells in a population over a period of time. There are different stages of the bacterial growth curve. Upon inoculation into a new medium, the bacteria shows 4 distinct phases of growth, that are the lag phase, the log phase, the stationary phase, and the death phase. Let us dive into each of these phases in detail. The first phase of the bacterial growth curve is known as the lag phase. When bacteria are introduced into a nutrient medium, they take some time to adapt to the new environment. Cells in this phase have to adjust according to the temperature, the pH, and the oxygen availability in the new environment. Before cell division, it is essential for the cells to repair any damaged cells and synthesize RNA, enzymes, growth factors, and other essential metabolites that might be missing from this new environment. The cells therefore increase in size, are metabolically active but are unable to divide, and therefore no cell division or cell growth occurs in this phase. The next phase of the bacterial growth curve is the exponential phase or the log phase. Once the cells have gathered all that they need for growth, they proceed into cell division. This is the time when the cells are dividing by binary fission and the doubling in numbers after each generation time. Metabolic activity is high as DNA, RNA, cell wall components, and other substances necessary for growth are generated for division. Depending on the type of organism, growth conditions, and the organism density, this phase, that is the exponential phase, can last for several hours. Cells in this phase are the healthiest and most uniform, which explains why in most experiments, cells from this phase are utilized. Next comes the stationary phase. Eventually, the population growth that has been experienced in the log phase begins to decline as the nutrient availability in the medium becomes depleted and the toxic waste products start to accumulate. The bacterial cell growth reaches a plateau or a stationary phase where the number of dividing cells equals the number of dying cells. This results in no overall population growth. Under the less favorable conditions, competition for nutrients increases and the cells therefore become less metabolically active. Physiologically, the cells become quite different at this stage as they try to adapt to the new starving conditions. Spore forming bacteria produce endospores in this phase, and the pathogenic bacteria begin to generate substances, for example, virulence factors, that help them survive these harsh conditions. Production of antibiotics such as penicillin, streptomycin, etc., by certain bacteria is also seen during this stationary phase of their growth. The last phase of the bacterial growth cycle is the death phase. This is the last phase of the bacterial growth. As the nutrients become less available and the waste products increases, the number of dying cells continues to rise. In the death phase, the number of living cells decreases exponentially and the population growth experiences a sharp decline. As the dying cells lyse or break open, they spill their contents into the environment. These nutrients are now made available to the other fellow bacteria.